then. Good morning, everybody, and thank you for taking the time to join us. This is our SEBA Expert Series, Seminar Series number 4 for 2022. And very happy to have with us today Dr. Tang Swee Seong. And he is a lecturer in the Faculty of Science, University of Malaya, as well as being our associate member in SEBA. And we're very glad to have him as one of the team. So just a little bit about him before he gives his talk. He got his bachelor's in biochemistry and master's in microbiology at University of Malaya. And then he went on to do his PhD at the Australian National University, ANU. That's in Canberra, right?
Yes, that's right.
Yep. Obtaining a PhD in molecular microbiology in 2013. And at the University of Malaya, he plays a key role as a PI for several research projects, and that includes isolation and characterization of bacteriophage against Shigella species, and elucidating molecular mechanisms and interaction of polyvalent bacteriophages against bacterial hosts. So if you want to work on phages, Dr. Tang is the person to talk to. And he's interested in developing bacteriophage cocktails for use in diagnostics, also for therapy against multidrug resistant pathogens in aquatic animals. And of course, that's a really important emerging area. One, to reduce the amount of antibiotics, but also because there are so many multidrug resistant pathogens emerging. And his ultimate aim is to develop natural alternatives to solve the crisis in multidrug resistance and the use of phages in the treatment of disease. So I'll now invite him to give his seminar, Good Virus versus Bad Bacteria, Phage Therapy Against Multidrug Resistant Pathogenic Bacteria. Dr. Tang, please present your seminar. Thank you.
Okay. Thanks for the kind intro, Prof. Jenny. Let me share my slide first, yeah. Can you see my sharing?
Yeah, that's very clear. Thanks.
Okay. Yes, I would like to talk about bacteriophage, a good virus versus bad bacteria. Phage therapy against multi-drug resistant pathogenic bacteria. As said, my name is Swee Seong. I'm honored to be invited to give a talk in this SEBA Expert Seminar Series 2022. Okay, let me start with the introduction. Phage therapy was originally introduced a century ago, almost 10 years earlier than antibiotic therapy. It has, however, never outperformed the later due to several reasons, which I will elaborate to you later. Anyway, a couple years ago, the highly renowned journal Cell has published an editorial notes giving emphasis on bacteriophages as potential antimicrobial. They mentioned bacteriophage as one of the 9 major research questions in microbiology. Okay, a bit of background about bacteriophage. Let's start with the questions. What is bacteriophage? What is bacteriophage? A bacteriophage is also known informally as phage, which is a virus that infects and replicates within a bacterium. Or in a layman term, a bacteriophage is a virus that eats bacteria. Bacteriophage or bacteriophages, they are composed of protein that encapsulated a DNA or RNA genome. They may have relatively simple or elaborate structures. Bradley has divided phages into 6 different taxonomy groups, A to F, as shown in the slide, based on their structural morphology and nucleic acid content, either double-stranded DNA, double-stranded RNA, single-stranded DNA, or single-stranded RNA. There are 19 families currently recognized as that infect bacteria and archaea, by the ICTV, International Committee on Taxonomy of Viruses. And about 96% of the total bacteriophages is from these three families: Myoviridae, Podoviridae, and Cephuviridae. Okay, why is phage treatment needed? In general, to treat bacterial diseases, antibiotics such as beta-lactams family, quinolones families, macrolides, and other antibiotics are commonly used. The gradual emergence of multidrug resistant bacterial pathogen has made these conventional antibiotics losing its magic bullet ability to kill bacteria. Multidrug resistant bacterial pathogens really impose critical challenge for clinical and pharmaceutical research due to their potentially severe impact on human health. The WHO is extremely concerned about worrying growth in microbial pathogens and antibiotic resistance in the world. This antibiotic resistance is caused by bacterial and social factors such as high mutation frequencies, coupled with the exchange of genetic information by bacteria, the misuse or overuse of antibiotics by human beings, and increasing population densities, and the global migratory movements by animals and people. The gradual emergence of multidrug resistant bacterial pathogen has indeed triggered the search for alternatives. Well, administration of antimicrobial peptides and antibiotics cocktails are promising replacements. However, this alternative may eventually suffer a similar fate as the current treatment for antibiotics. Phage therapy could be one such suitable alternative for its proven long-term safety profile. Well, a variety of phage-based approaches and products, including single phage, phage cocktails, mutated phages, genetically engineered phages, and combination of phages with antibiotics have already been carried out to test the application of phage therapy against multidrug resistant pathogen. All right, now let me tell you a bit about the early history of phage therapy. In the beginning of 20th century, 1915 and 1917, Twort and Deherrier independently discovered bacteriophages. In addition, Deherrier and his co-workers isolated phages with lytic activities against pathogenic bacteria, including Shigella species. And they developed the idea of phage therapy, meaning the prophylactic or the therapeutic use of these substances, the bacterial phages. Then the bacteriophages were then subsequently used in medicine from 1919 onwards, before the invention of the first antibiotic, penicillin. In the early stages, expectations were particularly high with regards to Shigella phages, the phage against Shigella, due to their success in treating dysentery patients safely. This success inspired the commercialization of therapeutic phages to treat bacteria infection in humans. However, at that time, scientists did not fully understand the mechanism behind the treatment, and in particular, how the phages kill the bacteria. Besides, the outcome of phage treatments were inconsistent. Moreover, the introduction of antibiotics in 1940s to treat a broader range of infections lead to reduction in phage therapy research. Despite the success of phage therapy in a number of Eastern European countries, it's remained largely neglected in Western Europe due to the inconsistent result, the lack of specific regulatory framework, and the complicated procedures for patenting phages. More recently, the interest in phage therapy has increased dramatically, and the use of phages in controlling bacterial infections has regained popularity, as well as those unanswered questions of phage therapy are now gradually being addressed. By looking back to the pre-antibiotic era, scientists aim to resurrect phages as antidote to antibiotic-resistant pathogens, as well as to solve other medical, agricultural, food safety, and environmental problems. Nowadays, the availability of those sophisticated molecular tools, the growing understanding of phage control techniques, and the evaluation experiences of Eastern Europe researchers have all widened the possibility of phage therapy applications. In Eastern Europe, phages have been administered orally, tablet or liquid, topically, rectally, intravenously for almost 90 years, with no serious side effects have been reported. As a result of this development, phage therapy has attracted increasing attention as a potential alternative solution to in treating antibiotic-resistant bacteria. Well, 6 clinics in 5 different countries, the US, the UK, the Republic of Georgia, Poland, and Belgium, are now offering phage therapy for treating diseases. There are 11 US and international biotechnology companies, as well as a number of academic investigators currently working in the field of bacteriophage technology and its products. Of course, I'm one of them. Yeah. These companies and researchers, they are utilizing bacteriophage or bacteriophages in the food processing industry and for the treatment of human diseases. For example, US companies such as Intralytics and Novalytics, they are using bacteriophages as biotechnology tools and as a platform technologies. The FDA, Food and Drug Administrations, and also the United States Department of Agriculture, USDA, they have marked a milestone in phage research by approving 3 phage products, each of them comprising a cocktail of phages to target and kill bacteria. Well, the first FDA-approved phage product was List Shell, which can be used as a food additive against Listeria monocytogenes in a ready-to-eat meat and poultry. The second product, EcoShell, which can be sprayed on the red meat in order to kill E. coli, Escherichia coli, before the meat is ground into hamburgers. The third phage product called Salmon Fresh, which acts against Salmonella enterica. It was approved as a food processing aid. It is used for the treatment of fresh and processed vegetables, fruits, seafood, such as shellfish and fish. Lately, another phage preparation, which is called as Shiga Shell, is currently undergoing FDA and USDA reviews for the GRAS status, generally recognized as safe status. The biotechnology phage company, Novolytics, that developed that one, has the aim to lead the utilization of bacteriophage as a treatment for bacterial infections. Currently, the company's most promising product, which is called Novo 12, is a phage cocktail administered as a form of gel for topical treatment. It can treat MRSA, the methicillin-resistant Staphylococcus aureus infections. Now, let's have a look at phage therapy versus antibiotic treatments. The differences between phage therapy and antibiotic treatments are as summarized in the table shown on the slide. Treating multidrug resistant bacteria by a new antibiotic or a new combination of antibiotics tend to be more complicated than treating it with a phage or a phage cocktails. Generally, phages are environmentally friendly. In both cases, the clinical trial is expensive, but it is usually quicker and less expensive to select, isolate, and identify phages than to develop a new antibiotic, which can take a longer period. Well, secondary infections may happen, but very rarely and minimal in phage therapy as compared to antibiotics treatment. The term secondary infection during phage therapy is due to the interaction between phage and bacteria, which can cause super infection immunity or super infection exclusion. With respect to antibiotics, it can cause secondary infection by attacking the normal flora of patients and the normal microflora of patients, in addition to the targeted pathogens. In addition, phage resistance is less of a concern than antibiotic resistance because phages can mutate and evolve naturally to counter phage-resistant bacteria. Moreover, the phage-resistant development can be mitigated by using phage cocktail. Phage cocktail is a combination of multiple phages, or it can be mitigated by applying phages in conjugation with antibiotics as therapy. One of the advantages of phages is that they have much fewer side effects. In fact, the prolonged use of phages to treat human infections in Eastern Europe has not elicited any allergic reactions, nor have animal trials in Western Europe revealed any unusual historical changes, mortality or morbidity when phage were administered either orally, intravenously, or intramucously. Indeed, the intakes of the T4 phage up to 10 to power of 5 PFU, the plaque forming units, have not caused any secondary effects. Okay, despite all the advantages of phage therapy, it is still a long way from being the magic bullet for treating infections, because many parameters like the frequency, the duration of treatments, the route of administrations, and the optimum dose, those parameters have yet to be determined precisely through clinical trials. Based on the reports from researcher groups, there are some major limitations of phage therapy as summarized on the screen. The first limitation is a narrow host range, as well as serotype specificity of phages, which might reduce effectiveness and coverage. Second, a single phage is inadequate for treating illness caused by multiple bacteria. Third, the release of various pro-inflammatory components like endotoxin, peptidoglycans from bacterialized by phages. This might cause problems in the human body. Fourth, there is a possibility that resistant bacteria might emerge after treating with phages. However, phages can evolve and adapt to combat this resistant bacteria. Fifth, complicated pharmacokinetics and pharmacodynamics of phage treatments and the interference by anti-phage antibodies. Well, in addition, there are other problems associated with patenting, manufacturing, administrations, which often create obstacles for development of phage therapy. The lack of definite regulatory outline reflecting individualized therapies, or difficulties for the pharmaceutical companies to register intellectual property, IP, for phage and phage products are some of the major problems in phage therapy too. The eventual success of phage therapy will largely depend on the development of appropriate strategy to overcome all these mentioned limitations. In addition, adequate regulatory framework must be created. Appropriate safety protocols have to be implemented, and general acceptance of public towards phage treatment is needed. Okay, for solution, several initiatives have been taken to overcome the limitation in phage therapy. Phage cocktails have been formulated in consisting of several phages with complementary features different receptors which can play a vital role to overcome the limitation of single phage with its narrow host range. In addition, phage cocktail containing different types of phage potentially capable of combating the same species and strains of bacteria could reduce emergence of bacteria resistant to phage. Moreover, the host range of phages can be broadened by engineering their genome to express endocellulase and by substituting the gene encoding putative host binding proteins. In addition, the challenge of phage therapy may be overcome by producing genetically modified phages, recombined phage genomes, site-directed mutagenesis, selected spontaneous mutants, or phage display techniques. Mutated phages could also be used to overcome phage resistance, as well as to prevent the human immune system against phages. The efficacy of phage therapy could be enhanced by utilizing the antimicrobial synergy between phages and antibiotics. So this synergistic effect could hasten cell lysis and allow phages to spread more quickly. Thus, antibiotic conjugated to phage could enable the delivery of antibiotics to specific cell and cause an increase in local drug concentrations. Well, in conclusion, phage therapies for pathogenic bacteria have been studied and applied for about a century, but phage therapy as an antibacterial treatment in general has not received much attention due to the lack of clinical knowledge, public awareness of phages. However, given that the development of novel antibiotics is laborious, time-consuming, costly, it makes eminent sense to seek alternative antimicrobial approaches to combat drug-resistant pathogen. While it inevitably has some drawbacks, phage-based biocontrol and bacteriophage therapy are very promising approaches to combat the challenge of pathogenic bacterial infections, particularly when the search for new antibiotics is stagnating. The potential of phage therapy have been acknowledged and revisited by many scientists over the last few decades. And there has been a rejuvenation of research into phage therapy. Moreover, phages have many unexploited potentials as an alternative to antibiotics, both due to the range of intrinsic variation of their mode of action, also due to the almost unlimited variety of phages and their ability to evolve in situ to successfully deal with bacteria resistant. Well, currently, phage therapy is encouraged in many parts of the world because policymakers consider growing MDR, multidrug resistant, as a serious health problem. This awareness should further encourage researchers to study the biological properties of phages, which eventually increases their safety and efficacy. Finally, cocktails of natural phages and genetically modified phages could open new perspectives for successful phage therapy in the future, particularly against major challenge of multidrug resistant bacteria. Here is the main reference I use for this talk. It's a review paper I wrote together with my research team. You may scan the QR code to download the paper to get more details. Lastly, I would like to acknowledge and Ministry of Education for funding me for the listed research grants to support my research regarding phage therapy. I would also like to say thank you to all my research students from the past to current, present, for joining me in the research related to bacteriophage therapy. And I would like to thank you for listening to my talk. I welcome collaboration. Here's my contact detail, and you can reach me. That's it. Thanks.
Okay, thank you, Swiss Young. That's a very interesting topic. I'm sure people will have a lot of questions, but I'll get started first. So my first question is, when you're working with this in the lab, what kind of biosafety or biosecurity permissions do you need? Is there any need for that? And how does it change if you wanted to use this in aquaculture? What sort of process would you have to go through to get approval for testing this?
The first question is whether any biosafety is needed, right? Well, actually for bacteriophage work, because phages are abundant, right? They are everywhere around us. So we don't even need the biosafety level to conduct this bacteriophage work, unless we are dealing with multi-drug resistant bacteria that need biosafety work. Okay, what was the second question?
So you were mentioning that one of the issues is to get approvals if you want to use these as products. So supposing you're developing it for aquaculture, like in Malaysia, are there any such products licensed in Malaysia? And if you were developing a new one, what is the process to get that permission?
What I know is if you are developing a phage cocktail product for human consumption, then we will need some kind of approval. But if for agricultural products, I don't think we need that approval.
That surprises me. I would think it should be under biosecurity, or there should be some kind of mechanism for that. Because in the spirit of like, how does it impact the biodiversity in ecosystems and so on, I would think when you're introducing large amounts of something, especially, I mean, if it's a product that was developed overseas, for example, it's not a phage that we have in Malaysia, how would that be?
Well, I think if we are engineering the phages, then we definitely need to have some kind of regulatory approval to use that. If it is the nature product that isolated in from local, so they are already there. So
yeah, but that is true. That's true for biofertilizers as well, but they still have to be regulated. You can't just use it. Yeah. No, it's why I don't know if we have Zatil or others here who are interested in sort of like the regulation, because I think if you haven't looked at that, that's another interesting area for a review paper. What is actually, is there any regulation, and what is the regulation, and how is it different in different countries? Because you said that they do need to be licensed as products, so there must be. Yeah, you should talk to others in SEBA. They might want to write a paper with you on that aspect. It just occurred to me because it's something different. if you were like adding a lot of phage to an aquaculture pond, for
in bigger
example,
amounts, right? Yeah.
it could have an ecological effect, and that might
That's
be
right.
something that should be regulated, or maybe is already regulated, or is anybody doing it and we don't know. I think that would be actually quite interesting. So
Yeah.
that was why I asked that question. Okay, I asked a lot of questions, so I will open it to others to ask questions. I think you are able to turn your mics on, so we have like who can shout the loudest gets to go because I can't see how to raise hands here. I think if you want to ask a question, or if you want to type in the chat box, I can also read out your question if you're too shy to turn on your mic.
I have one, Fikri here.
Dr. Fikri, go ahead.
Hi. Yeah. Thank you, Dr. Tang, for your presentation. It's really interesting and new information for me about phage therapy. So I see that you put a lot of examples of the application of phage therapy for like medical area concerning human health. I'm just wondering how is phage therapy applied in agricultural sector? Do you have any examples that you can share with us?
Yeah, I shared a few fish products just now. Some of them apply to the ready-to-eat meat, that is agricultural product.
If
You can spray on the vegetables.
oh, so it's applied on the produce, not on the crops. I mean, to protect them from diseases, um, bacterial pathogens that can cause diseases in the plants themselves. Is
Uh,
that is
yeah,
there any such application in terms of phage therapy?
I believe they are this type of application as
Mm.
but maybe the product hasn't really released yet, and we don't know about that. Like I'm working on bacterial fish product on agriculture, the tilapia fish. So I haven't got the product ready yet, so I haven't published them.
Yeah, okay. Because I'm just wondering how can you apply this kind of fish cocktail to what do you call it? A patch of land with crops. Do you spray them or do you introduce to the soil, stuff like that? Just interested in the context of crop um, introduction of the phage therapy to crops. Okay, thank you very much.
Yeah, if I can follow up from that question, because you mentioned you're doing it in aquaculture. How will you apply in aquaculture? Do you like inoculate the water, or you inoculate the food, or so the fish will eat it, or how do you introduce the phage to the fish?
At this moment, we are we are inoculating the fish into the water. Uh, we have a plan to incorporate the fish into fish feed, and also we plan to encapsulate the phages or fish cocktail, and then we could add it on to the fish feed, or we could put it in the water to let it release slowly. Yep.
And another question. Anybody else? Otherwise, I'm going to ask another question.
I have one probably.
That's Fadli.
Fadli
Yeah,
here,
go
yeah.
ahead. Go ahead.
For Jenny. Dr. Tang, thank you for the presentation. It's very interesting for me because it's new also for me. Regarding, you said something about the advantage of phages, right? They are able to mutate if the resistant bacteria are also changing. So these phages can mutate to accommodate to kill the new ones. But is there any risk of these phages to continue to mutate and and since there could be thousands of them, different phages in the body, and if they continue to mutate and kill off the other types of bacteria that we are not wanting to be deleted.
Well, because bacteriophage, they are bacteria eater. They only target on the bacteria. So we actually don't need to worry so much about bacteriophages. They are plentiful of them around us. In regards to the mutation they have against bacteria that mutated, they bacteria that may mutate to increase the antibiotic resistancy. There was a study about the mutation rate of phages against the mutation rate of bacteria, and they found that the mutation speed for phages against that particular bacteria is much more faster than the mutation for the bacteria itself. So they mutate only to let them able to eat that bacteria. So have I answered the questions?
I think so. I'm just thinking of any possibility for it to continue mutating, because they will be inside the body for a very long time, or they will be forever, right? Or they can be flushed out. I'm not so sure about that. Yeah. So I guess I am thinking of from the point of view of someone who doesn't know anything about this kind of treatment, and probably they might be worried about it, and we want to convince people that this is good, and so that kind of perception thing. So yeah.
Yeah, that's why I think for any phages that is being those phages that are going to be commercialized and to be used as a product, genomic screening for their existing genome for the safety gene or those things has to be done first.
Yes, I agree.
How
Thank you.
how host specific are they? So like if you were targeting, say you're targeting a particular bacterial pathogen, that phage, it can affect other species of bacteria, or it only will affect the particular pathogenic bacteria? Because I think this goes to the same question as like with antibiotics, we know that it actually kills all the bacteria, including the ones that are good for us. So it's always like after you've taken antibiotics, you may have some digestive problem because of this. So is it the phage is similar? Is it going to attack other bacteria as well, or only the one that you are targeting?
Theoretically, they are very specific.
Right.
Their specificity is even up to species and also serotypes level. Well, yeah, but there are also some exception, like the one that I've isolated. They are able to infect all the species, all the serotypes in that species. So they are kind of slightly broader in the host range, but still up to species level.
Okay, so does that mean, I think going back to Dr. Fadli's question then, does that mean that they will persist? Because as long as there's a host there, they will continue to be there.
That's right.
Or do they actually wipe out the whole population and then they die because they have no host? I just wonder how it's managed.
I think I don't know the real mechanism, but according to theory, if they have no food, they would be wiped off. If they have the host bacteria there, they will keep infecting, replicate, spreading, and then until they kill off all the food, and then when no food, they will.
So can any of these strains become lysogenic, or are they all lytic phages?
I'm not sure. I think if they could be possible for them to be lysogenic.
Yeah,
Yeah.
because I think it's just that I teach I teach phage biology. Sorry, so I know I have to teach this part, and that's why I'm interested, because if they become lysogenic, maybe there's more risk of them persisting, and maybe that is not desirable for the therapy. So I think I heard that mentioned before, that for therapy they try to not use the one that can form a lysogen. And then when it's lytic, if the host is not there, it will also go away. So it's easy to control. That's why I'm thinking there must be some kinds of regulations of what types of product can be used. But the narrow specificity is good, because that means they shouldn't affect the other bacteria. In fact, that would be better than antibiotics then, because you have a very specific target, which is good. Yeah, I think they have. I know they've been around a long time, but they are never quite taken off, right? Yeah.
Right. True.
Other questions? Anyone? There are no stupid questions, by the way. I happen to know about them, but if you don't, you can ask anything you like. Don't worry. Anybody now inspired to look at them? I think Dr. Fikri is thinking, maybe he should be using them for plant pathogens as well, because actually there's quite a few serious bacterial pathogens in our tropical plants. I know, like in banana, the wilt of banana is emerging as a problem. There are a lot of bacterial pests. So if they can be used in agriculture for crops as well as aquaculture, they might be very useful. Again,
especially if we have the the most simplest delivery system, like just direct spraying at the first symptom of infection, then it will be very convenient for the farmers.
There you are, a new proposal to write, Dr. Fikri.
Yes.
Together with
Need to delve deeper into phage therapy.
Yeah, you can write a new proposal for the next round. I think if it hasn't been done or it hasn't been done for Malaysian crops, I think it's actually something to look at. Yeah, there's potential there.
Quite a lot of potentials there.
Yeah. Yeah, because we must have loads of these things. It's like our bacterial diversity is so high, so the viruses must be also very diverse.
Yeah.
Yeah, I'm sure we have a lot that is unexplored. Any more questions? I'm trying to think. Microbiologists, we got Kawi here. You want to ask anything? He's very quiet.
So no question from me.
So far, no question. Okay, you can help Dr. Fikri write his proposal maybe, and with Sui Siong. I think that would be a really nice proposal to do that for plant pathogens.
That I plan to venture into that too later on.
Yeah, it's definitely worthwhile. Yeah. Okay, it's very quiet, so I guess everybody is already soaked up enough information, and they can always contact you, right, by email if they have more questions.
Yes.
Okay, everybody, if you can look at the chat, there is a link for our Google form. Please do give your feedback in our Google Forms for the SEBA seminar, what you liked, what you didn't like, let us know. And if there's no more questions, then I think I will close the seminar. Okay, it's just the form has just been repeated again in the text box, so please click on the link. It's very fast to fill in. It'll take you 2 minutes. Okay, so thank you again, Sui Siong, for this very interesting topic. I think we got lots of new ideas. That's really good. And and thanks everyone for attending today, and hopefully we'll see you again at another seminar soon. Okay, have a good day, everybody.
Thank you very much.
Bye.
Thank you.
Everyone.
Thank you.